Hello and welcome. My name is Sam Ankerson. I'm the director of the Bailey Matthews National Shell Museum and Aquarium. And thank you for joining us for this first of our off-season, hurricane season, whatever you want to call it, online lecture series. And we're excited to have Tim Pearce here with us to talk about land snails and some very interesting case studies that he's been working on. Tim is a um, longtime prominent malacologist and major contributor to the field. Has been at the Carnegie Museum of Natural History for some time, and before that, the Delaware Museum of Natural History. He is a good friend of this museum and a collaborator on an important project that's going on right now to document uh, distribution of eastern seaboard mollusks. Hey Tim, how you doing? And and we're glad to have him and also to have a program focused on land snails because particularly here in the Sanibel area, sometimes those marine mollusks get all the glory. And there's a lot of interesting work going on with land snails. And by the way, they often have really beautiful shells themselves. And tonight's talk, Tim will highlight um, some influences on species distributions, influences both ancient and modern. So we're looking forward to it. Before we get to the talk, just a couple of points. There are two more online lectures scheduled right now on September 19th. Myself and our curator and science director, Jose Leal, will be giving a talk. It'll be a preview on the new Great Hall of Shells exhibit here at the museum that will be opening a little bit later this fall. So that is in the very final stages, and we're excited to share that. And it'll be kind of a sneak peek on that. And then on October 17th, Gary Kidder from the Houston Museum of Natural Science will be giving a talk on carrier snails. So these are carrier shells, rather. So these are like tonight. They're like this talk. They're free, and you can go to shellmuseum.org to register. And while there, if you're interested, there's also an archive of somewhere between 20 and 25 um, videos of online talks we've had in the past. So you can peruse those and check those out. Check those out anytime you like. This program is in the webinar format. And so if you have questions, please use the chat function, which you can access by running your mouse along the bottom of the screen, clicking on chat and typing in your question. You can do that at any time. We'll handle the questions after Tim's talk, but please feel free to submit those anytime and we'll take a look at those and read them back and get them answered. And thank you in advance for your questions. And so now, a little biographical information on our speaker. Tim is assistant curator of mollusks at the Carnegie Museum of Natural History, where he manages a collection of 1.8 million mollusks. Prior to Carnegie, um, he was assistant curator of mollusks at the Delaware Museum of Natural History. His research, as you'll learn more about in this talk, focuses on the ecology and systematics of mollusks, biogeographical, ecological, and behavioral studies on terrestrial gastropods, and the inventory and distribution of land snails and slugs in the northeastern United States. He received his PhD from the University of Michigan and master's in paleontology from the University of UC Berkeley. And lastly, I'll just say something not on his official bio. Tim is an incredibly gifted and skilled teller of mollusk and snail jokes. And by me saying that doesn't mean we are putting pressure on him to give us any jokes tonight. But whether or not he does, I encourage all of you to go on YouTube, enter his name and mollusk jokes, and you will get a you'll get a great laugh and a great dose of these jokes, which are just, so bad they're good. So got to check it out. So thanks, Tim, for being here, and the floor is yours.
All right. Yes. Glad to be here. Um, Speaking of jokes, you mentioned that this is hurricane season, but there's not a hurricane currently. And so I thought, oh no, I should be talking about clams instead of snails, because then I could be the clam before the storm.
That's that's terrible.
But
Thank you.
so yes, I'm Tim Pearce at Carnegie Museum of Natural History with a presentation for you. And so besides telling jokes, I also do scientific research. And I'll be talking about two of my projects today. Um, One is about tiger snails and how they seem to be disappearing, and maybe we can explore why. And the other one is about how glacial history might have influenced which snails got to which islands in the Great Lakes. So let's see. I have control here. Yes. Um, so we are going to talk today about two snail projects. So could acid precipitation be a factor in the decline of the tiger snail, Anguispira alternata, in northeastern North America? And for the second part of the talk, we'll talk about glacial history and snail distributions. Did the glacial history influence which snails got to which islands? So for the first talk, first part of the talk, did the acid rain influence the decline of the tiger snail? So these tiger snails are endemic to North America. So that's the only place that they live in the whole world. And there are, I don't know, 15 to 20 species of Anguispira. And they are widespread. They're large and common. They are, roughly the size of a nickel or so. So they're pretty large. And so I used to say, hey, these snails are the most common large land snail in Eastern North America. But then over time, over the decades, I started noticing it was getting harder and harder to find them. So that's what brought up the question of why are they declining. But the fact that they are so common means that a lot of museum records exist. And museum records, so one of the hidden agendas in this talk is to promote museum collections. How can people use museum collections? Why should we care? And how are they useful? But the fact that many museum records exist means that there are lots and lots of records through time. So we can go back through time and find out when were they living in particular places. So back 8 years ago, I published, along with a colleague, an article about the decline of the tiger snail in Pennsylvania, and we suggested acid rain. So we considered four possible factors. Well, first of all, we demonstrated it really is declining, or it has declined, and we considered four possible hypotheses for the decline. One was deforestation. We looked at climate warming. We looked at invasive species such as earthworms, and we also looked at acid precipitation. So the timing, the snails seem to have started to decline around the middle of the 1900s, and the timing didn't work out for most of these hypotheses. The deforestation, that was mostly completed at the end of the 1800s, early 1900s, and the snail didn't start to decline till the mid 1900s. So the timing was off for deforestation to be the culprit. For the earthworms, oh, by the way, those of you who think that earthworms are amazingly good things, they're good in your garden, but when they get out into the wild, they can actually cause a lot of havoc. And just as an example, imagine that you are a rare plant and you love to live in poor soil. And then the earthworms move in and turn the soil into good soil. Then you're no longer a happy plant. And so earthworms can be bad for the biota where where earthworms don't belong. So they're good in your garden, but earthworms are actually causing a lot of problems. So the earthworms actually started moving out of our urban areas into the more rural areas around the end of the 1990s, 1980s, 1990s, which again, the timing is off. So the snail was declining in the mid 1900s, the earthworms were becoming a problem, an issue in our natural areas in the much later. So the timing is off there. For climate warming, at least back in 2016 when we wrote this paper, climate warming, I was under the impression that that wasn't really the signal wasn't rising out of the noise until again 1995 or end of the 1990s. And again, so the timing is off. Since then, though, I have learned that the climate warming was having effects much earlier. So actually, climate warming could be a factor. And I'm not prepared to talk about that very much, but we can talk about it during the discussion if you like. But the thing that did fit timing-wise was acid precipitation. So acid precipitation really started taking off in the middle of the 1900s, and that's about the same time that the snails started declining. So that's why we suggested acid precipitation for what was going on in Pennsylvania. So today, I want to revisit the acid precipitation hypothesis and expand with a much larger data set. So we're going to include 26 states, counting DC as a state, in the whole northeastern United States. And in addition, we're going to consider search effort and calcium. So let's go get on with that. So these two maps show that the blue counties in these maps are the counties where the snail is known to occur. And if you look at the left map, before 1960, the snail occurred quite a few places, or it was known to occur in quite a few places. In the map on the right, the counties, these are counties since 1960 where the snail has been seen. And it's quite a bit less. In fact, it's a little fewer than 50% of the counties where it is known after 1960. So when I first saw that, I thought, maybe the people after 1960 are just lazy and they're not looking hard enough. And so I actually looked at it for a couple of years trying to convince myself, are we just lazy nowadays or are we actually looking hard enough? Well, in the next slide, I'll get into one of the ways that convinced me that, yes, the decline is real. But these maps are using almost 5,000 records of the tiger snail, Anguispira alternata, from at least 30 different museums. And so is the decline real or are we just lazy butts in the modern times? So this graph shows all of the snails, not just the tiger snail, but all of the snails in all of these 30 museums shown by decade. So let's see, I think you can see my pointer. Yes. So in 1890, there were, I don't know, almost 3,000 total lots of snails, total records of snails collected during that decade. And so you can see between 1890 and I don't know, about 1980 or so, there were, I don't know, what is it average? Maybe it averages around 5,000 records of snails per decade. But if you look from 1990 onward, it actually does go up. So there are more snails being found in the more recent decades, which kind of convinces me not only are we looking hard enough, we're looking harder than the people before. So that's one of the things. That's one of the two things that convinced me that we are looking hard enough. The other one was the species accumulation curve. And as you get closer and closer to finding all of the species in an area, your curve levels off. And so it was starting to do that. And so that was the second thing that convinced me. So I think that the decline is real. So now, as far as the decline goes, these bars show all of the snails in these 26 states by decade. The top bit of the bars, the blue bit of the bars, are the tiger snails. So, for example, in 1890, there were nearly 3,000 records of snails, and a few of those were tiger snails. So it's hard to see how wide the tiger snail slice is on those bars. So I'm going to take that slice and turn it into a graph here. So again, in 1890, what percent of all the snails that were, of all the snail lots that were brought in, what percent of those were tiger snails? And it's just over 5%, which means in 1890, when you went out collecting snails, when you brought them back, 1 out of 20 snails was a tiger snail. So that's not too bad. In fact, it looks like the percent stayed pretty, and it went up and down over the decades, but up until the mid 1900s, it stayed pretty constant at roughly 5% of the snails were tiger snails. But then starting the middle of the 1900s, so from 1950 onward, there seems to have been a drop, and it's gone down to maybe averaging about 3% of the snails that were brought in during any collecting event were tiger snails. So instead of 1 out of 20, it was more like 1 out of 30 or 35 snails would be a tiger snail. So I think that this is kind of convincing that there really is a decline and that it might have started happening right around the middle of the 1900s. So these maps, the blue maps show again the same maps that I showed before, that before 1960, there were a lot more records of snails in counties than there were after 1960. And so I'm curious to know, how does that relate to the acid rain? So up here in the upper right, I'm showing an acid rain map from 1985. That's the longest ago acid rain map that I could find. And you can see that here in the Northeast, we've got quite a bit of acid rain. But up there in the Minnesota area and Wisconsin, there's not quite as bad acid rain back then. And so the idea is that in areas that have more serious acid rain, if acid rain is the culprit causing the decline, then in areas that have more acid rain, you would expect more of a chance of that the snail would disappear in the counties. So if the county has more intense acid rain, there's a greater chance that the snail will decline in those counties. So let's take a look. Well, actually, first we need to, let me back up one bit here. So I need to consider search effort. And just as an extreme example, let's suppose that there was a county where the snail was known before, like let's say, I can't remember what county that is, but there's a county in Minnesota where the snail was known. Whoops, sorry. Oh no, what have I done? All right, here we are. Let's see, where's a good one? Okay, this county down here in Missouri. So the snail, no, that's a good one. Here, this county over here in Missouri, the snail was present before 1960. What if nobody even looked after 1960? If nobody looked, then there's no records, and I would naively say, well, the snail has disappeared. But that would be a fictitious record, because nobody looked, so we can't even say whether it declined or not. So that would be unreliable to score that as a disappearance. So what I need to do is figure out how much search effort is enough search effort to find it if it's really there. So I looked in the counties, and I looked at counties that had 10 records of any species of snail. And if they had the tiger snail in it, sorry, if a county had 10 records of any snail, 50% of them reported Anguispira alternata. If the county had 50 records of any snail, then 80% of those counties had Anguispira. And if you want to get your 95% confidence interval, that's what we use in statistics. It took 118 records in a county to be 95% confident. Well, if I were to use 118 records and throw away all the other counties, I would throw away most of my data. So I'm going to go with 50 records and say, if a county has 50 records after 1960, then I believe that that's enough search effort that I'm going to believe the absence. If alternata was not found, I'm going to believe that it disappeared there. So again, if the snail, Well, if the snail is present after 1960, then of course we know it persisted. And by the way, there are instances where it was not seen before 1960, but it was seen after 1960. So I'm going to count that as a persistence, even though we don't have a record of it before, because I believe it was there before. It was just overlooked. I don't think that the snails are migrating in just in a few decades. So if it's present after 1960, we're going to call it a persistence. But for disappearance, it has to be not found after 1960, and it has to have been found before 1960, because you can't have it disappear if it was never there in the first place. And it has to have enough search effort. So it has to have at least 50 records of any snail after 1960. All right, so this map is kind of combining all three of those bits of data. So we've got our presences, and then we've got I've got our presences in blue. So persistences in blue. Our disappearances are in red. So these are counties that had enough search effort and it was present before, but it's absent after. And then the white counties are counties where either it was never present or there's insufficient search effort to feel confident that it had disappeared. So what you can see in this map is that, hey, look, it's mostly in the east where we had the more serious acid rain. So it sounds like it's getting consistent with our hypothesis. You can see the serious acid rain sort of coincides with the places where there was more disappearances, where there were more disappearances. And in fact, it is a significant difference. Where there was more acid precipitation, it had a greater chance of disappearing, and that was pretty highly significant. So that's consistent with the idea that acid precipitation is the cause of decline. Now, the astute people among you, the observant people among you, might be saying, hey, what about Ohio and Indiana and Michigan too, and maybe Illinois and Kentucky? Look, they've got serious acid rain, but they don't have a lot of disappearances. I hope that was you. I hope you were that observant person who noticed that. Okay, so let's take a look at that. I think it might be calcium. So here's a map of soil calcium. So if you don't know land snails love calcium. If you ever want to find a place with lots of land snails, go to a place that has lots of limestone or an abundance of calcium, because snails just do really well with calcium. So if you look in these maps, Ohio and Indiana and Illinois, maybe the southern part of Michigan, I'm not sure about Kentucky, but they have quite a bit of soil calcium. The red and the yellow areas have more soil calcium there. And the calcium abundance increases farther to the west. And so that kind of matches up with what we see with the snail disappearances. Snail disappearances are less in the areas that do have more calcium. So it could be that the calcium is acting kind of like a buffer. And so if there's acid rain, then the calcium might be absorbing the acid and allowing the snails to persist there. And in fact, yes, that was a significant difference, barely significant, but it was significant that there were the tiger snail persisted more where there was more calcium. All right, so it looks like acid rain probably was an important factor in the decline of the tiger snail. Now, we humans have done a pretty good job of cleaning up our act. We've really cleaned up our power plants. A large part of our acid rain came from power plants. We've done a lot of cleaning up of our power plants and other sources of acid rain. And so you can see these maps. The left one is 1985 and the right one is 2012. So it's cleaned up quite a bit. There's still a little bit of acid rain here in the east, but it's a lot better. So you might say, hey, maybe the snails can come back now. And that's a possibility. But one of the things that happens with acid rain is the hydrogen ions get into the soil and they bump off the calcium ions from clay particles and things. And then the calcium ions wash downstream. So they're taken out of the system. And so even though we've cleaned up our acid rain, it's going to take decades, if not centuries, for new calcium to weather out of the rocks. So I do think that the snails will come back, but it might take a little bit of time. All right, so in conclusion about the tiger snail, the populations of Angospira alternata did decline, and the decline seems to be real. And there was greater decline with more severe acid rain, less decline when there was more soil calcium. And my plug for museums. Museum data are useful for addressing interesting questions over time. All right, let's move on to the other part of the talk, talking about the glacial history and how that might have influenced the distributions of snails on Michigan islands. So the land snails on the islands are, or historically have been, pretty poorly known. There are more than 560 islands in the upper three, in the northern three Great Lakes, but there are publications talking about the land snails on only 8 of these islands. What's going on? And only 3 of those publications talk about snails in Lake Michigan. Actually, one of them is here in the Beaver Island archipelago. So we'll be talking a lot about the Beaver Island archipelago here. So here is a closer up view of the Beaver Island archipelago, which extends here from South Manitou up to Hog Island. And I'll give you some results. We just got them in from Gull Island. That's what sort of got us started asking about glacial history. And it's sort of surprising. Oh, so South Fox is the only island that has had, only island of the Beaver Island archipelago that has had land snails presented from it. And it's surprising that there are so few publications on land snails because these islands are popular tourist destinations. There are permanent human habitations on Beaver Island and on South Manitou Island with, well, let's see, I think daily ferry service from South Manitou and at least twice a week on Beaver Island. No, sorry, daily on Beaver Island. I think daily on South Manitou too. So it's easy access. Why haven't more people published on the snails here? I'm not sure. So I'll give you a few results from Gull Island, which is the most far out of the Beaver Island archipelago. And it is also one of the smaller ones. And it's a wildlife refuge. So it's only 2.3 kilometers long, or for those of you who speak English, it's 1.4 miles long. And the geology is glacial till, so it's underlain by limestone, which is good for snails. And in fact, there are plenty of snails out there. Vegetation includes sugar maple and yew and some other trees. The yew plants mean that there are no deer currently on the island. Deer love to eat yew plants. So that's one way you can tell whether there's a deer population. But it also means it's hard getting through there because the yew plants grow really thickly together and it's difficult walking around. And there's a lot of shoreline, and as I mentioned, it's a wildlife refuge. Lots and lots of birds are nesting, especially down here in the southwestern part of the island. For methods, we went to the various locations on the islands and collected leaf litter samples, and then dried and sieved and picked out the tiny snails and identified them. Now, 2 of the species on the island are as big as a penny or as big as a nickel, but a lot of them are much smaller. And not all of these species live on Gull Island, but a lot of them do. And here you can see Punctum minutissimum is the size of Lincoln's nose on a penny. So there are a lot of snails that are really pretty small on the island. So sieving is really necessary to recover all of the species there. We tried to get good distribution from across the island, but sometimes we had limited time there. We got there by float plane, and sometimes we had limited time. So we did get a good sampling of various locations, and 18 localities. And if you look at the different species, some of the species they varied in abundance. So the tiniest snail out there is Punctum minutissimum, and it was very abundant. We found more than 1,200 individuals, whereas the some of the less common ones, Paravitria multidentata and Striaturifera, they were much less common. The two large ones, Anguispera alternata, they were not particularly common, and Neohelix albolabris, even a little bit less abundant, I should say. So that's as far as the abundance of the different snails go, 24 species. But from the 15 localities where we have good data, where we have leaf litter samples, it was sort of surprising. Well, let's see, none of the species occurred at all 15 localities, but most of the species occurred at most of the localities, which I thought was pretty interesting. It might be a reflection of the fact that it's a small island, so snails can probably access most of the different localities. But there were some, like the Striaturifera and the Paravitria multidentata, that were not very, did not occur at very many of the localities. I am happy to say that all of the species out there are native species. So that gives me hope for the future. It's probably a pretty pristine island. And the fact that it's a wildlife refuge probably helps for it to continue being pristine. Another interesting thing to look at is the species area curve. So species area curve is a well-known phenomenon, probably the best phenomenon in ecology, that shows if you look at a larger area, then you get more species. So that's the species area curve. And when you apply that to islands, you should get the same pattern. So here on the x-axis, we've got island area with the larger islands on the right, and then we've got number of species on the vertical axis. So I've eliminated the three largest islands that were actually not thoroughly sampled. So they probably would have more species than they're reporting. So I'm just focusing on the 5 islands that have been thoroughly sampled. And you can see that it fits the species area curve. More species as the island size gets bigger. And the smallest one here is Gull Island that we were just looking at. So it has the fewest species, and the species are ranging from 24 up to 45. And so there's a theory called island biogeography theory. And this theory predicts that you will have fewer species on smaller islands and fewer species on islands that are further from the source of dispersers. So fewer from the mainland or fewer from another large landmass. And so Gull Island fits both of those criteria. It's the most far out of the islands, and it's also one of the smallest ones, or the smallest one of the ones that we've studied. So it is consistent with island biogeography theory. So it's a well-behaved island. Yay! Thanks, Gull Island. But we also noticed that some peculiar distributions on not just Gull, but on the islands that we've looked at. We saw that the snails that were on the islands looked more like the snail faunas on the Upper Peninsula than they did like the snail faunas on the mainland. It was like the ones on the islands were a subset of the ones on the mainland. And as you go from island to island, it was often the same species were missing. And I thought that was kind of peculiar. And the fact that they looked kind of like the Upper Peninsula made me think, I wonder if it had something to do with the glacial history. So here are some maps that depict the glacial history. So if you go back, let's see, I think that the glacial maximum was 18 to 20,000 years ago. Here we are at 16,000 years ago. And so this map is a little hard to read, but the whitest areas there are a glacier. And so this is miles thick of ice, or kilometers thick of ice. And so there are no snails living underneath the glacier. And so here in northern Michigan, there are no snails living there. And on these, what will become the islands, there are no snails living there either. And then 11,500 years ago, the ice is still covering northern Michigan and the islands. But I'm proposing, my hypothesis is that the cold adapted snails are following on the heels of the glacier. So they're moving northward. Remember, this is happening over thousands of years. So I think that they actually had time to walk or crawl. So the snails, the cold-loving snails, are on the heels of the glacier. And then here in the lower left, 9,500 years ago, we've got the ice has retreated into the upper peninsula. And so on the lower peninsula, we've got land that snails could occupy. And look at that. The proto-islands are connected to the mainland. There's a land bridge. The snails could actually walk there to these proto-islands. And then 3,500 years ago in the lower right, the lake levels rose and cut off the islands from the mainland. So the land bridge is gone now. So trapping the cold-loving snails there. And then when the warm-loving snails finally moved in, they had a harder time getting to the islands. There was no land bridge there. So if my scenario is true, if my hypothesis is correct, then what you would expect is that the snails on the islands would be cold adapted, and the snails on the islands and on the mainland would be cold adapted. And the snails that are on the island, sorry, on the mainland only, not on the islands, those should be the more warm adapted snails. So let's take a look at to see how that works out. So here, first, I'm going to look at just the 4 largest species. So here we've got Anguispira alternata. That's the same species we talked about in the first half of the talk. Neohelix abolabris, Haplotrema concavum, and Mesodonthyroides thyroidus. And if you look at these maps are from Leslie Hubrick's 1985 Atlas of the Land Snails of Eastern North America. Anguispira alternata is a little bit farther north in the United States, as is Neohelix abolabris, suggesting that they might be a little bit more cold adapted, while Haplotrema concavum and Mesodonthyroides thyroidus, they're more southerly distributed in North America, and so they might be more warm adapted. And so all 4 species do occur on the mainlands. Okay, that's not a surprise. But check it out. Anguispira alternata, mainland and islands. Neohelix abolabris, mainland and islands, which, as I predicted, those should be the cold adapted snails. And then Haplotrema concavum and Mesodonthyroides thyroidus on the mainland, but not on the islands. And I predicted the more warm adapted snails should be absent from the islands. Now I've got an asterisk here by Mesodonthyroides thyroidus because it actually does occur on 2 of the Beaver Island archipelago. So I have no good explanation for why it occurs on those islands. Could have been brought by humans, could have, make up your story, but that's arm waving. But anyway, it does occur on 2 of the islands, but it's absent from the other ones. And still, even with that little bit of not 100%, it still is consistent with the hypothesis that the warm-loving snails got there too late to make an easy passage to the islands. So next, what I'd like to do is to add small snail species into the mix to see how they pan out. And I'm expecting it to be a little less clear because small species of snails, they can probably migrate even over water. They might be able to ride on birds. They might be able to be blown in the wind. They might be able to float on debris floating across the water, or they could be carried across by humans, unbeknownst, because they're so tiny. and there have been Native Americans out there for many years before the Europeans got there. So I expect it to be a little bit less clear. And so again, we're going to use as our proxy of cold tolerance, I'm going to use a proxy of the northernmost distribution range of the snails. So here's a couple of examples. For Vitrina angelicae, and these maps are from GBIF, the Global Biodiversity Information Facility, which takes information from, again, participating museums, yay museums, and it aggregates all of this information so that so researchers can make use of it. So I looked at the map of Vitrina angelicae, and I looked at the northernmost point where it has been reported, and it was up there at 56.1 degrees north. So I'm using that as a proxy of cold tolerance. And for Glyphylaena rhodesi, the northernmost one that I could find was 45.9 degrees north. So I would say that Vitrina angelicae is more cold adapted than Glyphylaena rhodesi is. And by the way, since I and some of the people there at your museum have been involved in georeferencing, I want to report that not everybody georeferences correctly, especially if you're using a robot to do it. And so in many of the maps, I looked at the northernmost point and said, do I believe that? And so then I actually went to the museum's record and read the locality description, and I said, that's not way that far north. They misplotted this one. And so I have actually reported to some of those museums and said, I think you need to move this one farther south. So the ones that I did choose are ones that, yes, I do believe and I do trust. I didn't just trust the dot. I actually went to the museum and verified that the description, the locality description was good. All right, so let's take a look at how it pans out. So if you look at here on the left graph, if you look at species that occurred in the mainland and anywhere from 1 to 13 islands, actually there were never any on one, just one. So from 2 to 13 islands, those had a range, but they extended much farther north than the ones on the mainland only. So I interpret this to mean that, yes, the ones on the islands are more cold adapted than the ones on the mainland. But like I was saying earlier, some of the snails could have gotten to the mainland much more recently. And so what if we just said, okay, well, let's say if it got to 4 islands, that's okay, we'll still consider it to be mainland. And so here I'm in the right graph, I'm comparing mainland plus anywhere from 5 to 13 islands. And then on the right side of that graph, we're saying mainland and anywhere from 0 to 4 islands. And it still comes out significantly different that the ones on the mainland and the islands are more cold adapted than the ones on the mainland. So I think that that's pretty convincing evidence. It's certainly consistent with the hypothesis, but I think it's pretty convincing evidence that the snails were influenced by the glacial history. Another way to look at it would be to say, well, so of the cold adapted snails, how many of them occur, or how many islands do they occur on? So you would expect a cold adapted snail to have been able to get to more of the islands. And in fact, we do see it. This graph has a lot of variability on it. So here on the x-axis, we've got latitude. So how far north did they go? To the right is farther north. And on the y-axis, the vertical axis, it's how many islands they are known from. And so the least squares fit curve, the dotted line there, is going up, and it is a significant curve. So yes, they are snails that are more cold adapted do occur on more islands. So again, that's fitting with the hypothesis, and I think it's pretty convincing evidence that the glacial history did have an influence on which snails got where. So in conclusion, for the glacial history part of the talk, Gull Island does have land snails. It's a small island and isolated, and it has the fewest of all the snail species. So it's consistent with island biogeography theory. Yay, Gull Island. And also then the glacial history has likely influenced which snails got to which of the Michigan islands. The island snails tend to be the ones that are more cold tolerant, and those that are more cold tolerant occur on more of the islands. All right, so I've got lots of people to thank for the tiger snail part of the talk and for the glacial history part of the talk. I do want to point out here the bottom acknowledgement for the tiger snail talk is, thanks to the Clean Air Act of 1970, we have reduced acid rain. So it has helped us to clean up our act. And I'm grateful to the float plane driver, pilot, Don DeRitter, to help us get out to the islands. And here's a cartoon for you. And then I will be happy to take some questions. The cartoon reads, It's a grandfather snail talking to the young ones. And it says, then suddenly, whoosh, the glacier came out of nowhere and overtook all but the speediest of your ancestors.
Which ties in perfectly with part of your talk. Thank you, Tim. I have to admit that that during some of the slides on the tiger snail distributions, it being an election year, and all those maps with the blue and the red and the
I
swing
didn't even
states,
think of that.
maybe you could consider a study of tiger snail populations relative to swing state counties.
or or tiger snail politics.
Tiger snail politics, right? Okay, I know we have some questions here. I know there are a couple from Lynn. All right, we'll get right to it. Okay. Lynn asks, do you notice a size bias in what snails are reported? She says, my own experience with marine snails is that the small fauna are much less likely to be reported.
Um, well, right. In museum records overall, there are definitely more of the larger snails. So on the snails that I've been talking about, these, well, with one tiny exception, the ones that I reported are just the ones for which there were leaf litter samples. The one exception is South Manitou Island. That's the farthest south of the Beaver Island archipelago. And that one, I did visit there, but I visited there before I realized that leaf litter samples are necessary. So that one doesn't have any leaf litter samples. And so I could have excluded it from my study, but I didn't want to because that's one of the two islands where the Mesodyne thyroides occurred. So I wanted to keep it in there as more of a rigorous test of the hypothesis. If I can show something's going on even by keeping Mesodyne thyroides in there, then maybe something really is going on.
Mhm.
So yes, I only included records or situations where there was definitely enough leaf litter samples so that I could eliminate the size bias.
Thank you. And Lynn had a follow, or I don't know if it's a follow-up question, a second question. Have you used the paleobiology database to see what's been reported for the Pleistocene sediments?
No, that would be very interesting to do.
Oh,
I will make a note of to do that.
all right. Thanks, Lynn. Our own Jose has a question. Are there any invasive snails in the study area that could exacerbate the acid rain damage done by outcompeting the tiger snail?
Oh, one thing that I forgot to mention was when I was showing um, the proportion. So the proportion of the tiger snail went down relative to the other snails.
Mm,
So a huge assumption that I forgot to mention is
mm,
I assumed that the other snails were not being affected by the acid rain.
right,
That's probably not true, but it
right.
is an assumption that I made. Um, so instead, I guess I could say that the tiger snail was affected more than the other snails were.
Mhm.
So Jose's question was,
Question was, are there any invasive snails in the study area that could exacerbate the acid rain damage done by outcompeting the tiger snail?
um, I think so. So in areas where I did find the tiger snails typically occur in good forests, and so there are fewer of the invasive snails in there, except that there are quite a few invasive slugs. So the
Mm,
Arion slugs, Arion subfuscus and fuscus, those are out there. I have no idea if they could be competing. I suppose it's possible.
yeah.
Um, they their populations exploded here in the Northeast um, in the late 1980s and the early 1990s, which I think that's a little bit late to be explaining the decline of the tiger snail.
Mm,
But that's a fascinating question. I'll have to
mm.
look more into that.
And I don't know why, but that your response to that prompts a question sort of I was thinking about during your talk is as far as the methods current, the contemporary methods for um, documenting populations of land snails. What are they? I mean, does iNaturalist play much into this, or is it what's the methodology for actually, the observations that feed the records?
So yeah, I think I mentioned that I was using records from iDigBio and Invert eBase, and at least one of those, if not both of them, do use records from iNaturalist.
Yeah.
As Lynn mentioned, or she asked about the size bias. So iNaturalist is definitely size bias for the larger species.
Mm.
So there would be more records from the iNaturalist records. There would be more size bias for the large ones, but there are quite a few, and like I said, the samples that do have leaf litter samples, those would um, definitely include a lot of the tiny ones.
Yeah.
But yeah,
Okay.
I'm looking for records from anywhere I can get them.
Anywhere you can get them, right? All right. Here is a question from Carol. What are some identification points we would need to know to identify this species? I'm not sure which species she's talking about.
She must be talking about Anguispira alternata.
Okay.
So yeah, you probably noticed that the southern limit was the bottom of Virginia and Kentucky and Missouri, and that's because there are so north of there, Anguispira alternata is the primary species. There are a few that occur there. Below that, there are records of Anguispira alternata, but there are some other species there too that have been misidentified. So to avoid any of these misidentification problems, for example, Anguispira fergusoni, it's more of a coastal plain species. That does not seem to be as affected by acid rain as Anguispira alternata. So I wanted to avoid misidentification confounding factors in my study. So I cut off my study at the bottom edge of Virginia.
Hmm.
You probably will have Anguispira in Florida, but I'm not sure that you would have Anguispira alternata. Um, I'm not sure what features to tell you. So look at how strong the ribs are on the shell. Look at the spot patterns. Look at the
Hmm.
the shape. Is it carinate or is it rounded? So there are a bunch of features that you can look at. And if you've got a copy of Henry Pillsbury's 1948 monograph, that's got a lot of good features for how to identify the different species of Anguispira.
Hmm. Thank you. All right. Here is a question from Marla. She says, that's amazing that Gull Island has no invasive snails, which actually amazed me too when you said it. I mean, it just seems like invasive species are everywhere, and there's still some place where there's not. So that's great. And then she asks, but were there invasive earthworms on Gulf Island?
Gosh, it's been decades since I was there. I don't think that there were any earthworms. I don't remember seeing them.
Yeah.
I can't say
Okay.
for sure, but I don't think so.
Yep. Another question from Jose. Any data on possible increased availability, sorry, any data on possible increased ability of the island snail species to withstand immersion in water, which could make it easier for them to, quote, float and passively migrate from the mainland to the islands?
So you're suggesting that the snails that made it to the islands might be more water tolerant? That's a very interesting question. I have not looked at it. Um, I guess it's something that I could look at. So one of the advantages of the islands would be these are in freshwater. So in Florida, where you are, it would be much more difficult for a land snail to float from one island to another because snails, as you know, are usually not very, land snails are usually not very tolerant of salt. But in the Lake Michigan Islands, and that's partly why I was expecting a messier distribution pattern from the smaller snails because they could have floated. And there are times, I know along some of the shores of the Great Lakes, there are times when some of the shore drift is full of land snails, including live ones. So that suggests that they can tolerate floating in the water for some period of time. But I'm not sure. That's an interesting question. Would the ones on the islands be more tolerant or survive immersion in water longer? I'd have to look at that. That's an interesting question.
It's a question for another day. Yeah.
Thanks, Jose.
Yeah.
Hey, maybe
Yeah.
we should collaborate on that.
Yeah. And thank you, Tim, very, very much. We were talking before the program about the how important it is in whatever ways we can to raise awareness about this group of animals and their place in the systems. And you are a wonderful communicator and ambassador on the subject. So thank you very much for being part of um, our lecture series and presenting this program. And there aren't any more questions, and we thank you very much
Well,
for all of it.
in
Yeah.
addition to your promoting, advancing our knowledge of mollusks, I also want to say, and museums. Museums
Yeah,
are really
right.
important as sources of information for researchers.
No, you're right. And thank you for mentioning that a couple times in your talk. It's absolutely true. Yeah, the museum collections are play an important role in all this. So thanks for your work on that too. I mean,
And
I know there's a lot of it.
if you haven't had enough snail jokes, here's one. Jose might have heard this one yesterday, but some snails were standing around talking about coincidences. And one of the snails said, "Hey, my wife was reading Ali Baba and the 40 Thieves, and she laid 40 eggs." And the second snail said, "Wow, what do you know? My wife was reading 101 Dalmatians, and she laid 101 eggs." And the third snail said, "Oh no, my wife has been reading 2,000 Leagues Beneath the Sea."
Bravo.
Better start saving for their college education.
There you go.
20,000 babies.
All right. Thanks, Tim.
All right. Thanks,
Thank
everybody.
you, everybody, for joining. Thank you very much, Tim. And we'll see you all down the road. Thank you.
All right. Bye-bye.